all of you. I am P.S.N.Darshanie and today I am going to present the research on conservation breeding of Mystus nanus, optimization of hormonal induction for spawning success. Currently, there are 4 types of Mystus species are recorded in Sri Lanka. Within that, Iriangutta, which is also known as strip-draft catfish, and is scientifically known as Mystus nanus, are recently found as an endemic fish to Sri Lanka. As you can see in this map, this fish is found in lowland streams and rivers. They have demersal, nocturnal and omnivore behavior. Apart from the morphological features of the Breeding family, they have unique features to identify. Those are the black hyperal blotch in their caudal peduncle and 2 green strips along their flanks. These fish also play a major role in our ecosystem, as well as they are very important to our economic value. Even though this fish is considered as the least concern in IUCN in 2020, now they are facing threats from habitat loss and pollutions. That means they have risks of extinction. But currently there are no any information about their captive breeding programs for the conservation. To conduct this research, all the ethical clearance were obtained from the Ethical Review Committee of Faculty of Applied Sciences, Rajarata University of Sri Lanka and Department of Wildlife Conservation. The general objective was to determine the best hormone for successful induced breeding of endemic fish, Mystus nanus. To achieve the general objective, the following specific objectives were focused. When it comes to the material and methods, total 27 males and 13 females were collected from the Kalawaba spillway using scoop net during night times. Then they were carefully transferred to the Nature Center, Dambulla. The brood stock selection was done by observing the presence of the genital papillae in mature males and genital core in mature females. Then they were kept separately in 2x2 concrete tanks for the 1 month acclimatizations, and as the feed, commercial feed was given at ad libitum. There were 3 hormones used for the treatments. In experiment 1, ovulin, which is gonadotropin releasing hormone analog with domperidone, and in second experiment, luteinizing hormone releasing hormone analog with domperidone, and thirdly, human chorionic gonadotropin hormone were given. Apart from that, natural breeding was conducted as a control. These all experiments were performed in 3 replicates. Then, the hormone doses were administered to the weighted brooders intramuscularly using sterile syringe at 45 angles. Then, they were introduced to properly arrange harpas as a sex ratio male to female 2:1. Then, the harpas were covered with the net to prevent escaping the brooders. After the spawning, immediately brooders were removed from the harpas and breeding success was determined. Complete randomized design was used as the experimental design, and as the statistical analysis, the mean values and standard deviation calculated using MS Excel and one-way ANOVA using Minitab software. For the estimation of the breeding success, relative realized fecundity, rate of the fertility of the eggs, rate of the hatchability of the eggs, and survival rate of the larvae were determined as the following equations. The courtship behavior commences within 3 to 4 hours after the hormonal injection. Then males exhibit the actively chasing of females. As the success of the induced breeding, the hormone ovulin resulted as the success in all the 3 replicates. while other two hormones success in only one replicate to each. Natural breeding did not occur. The type of hormone is not significantly effect of the breeding performance. This variation may be due to the environmental changes and handling stress levels such as two doses application and natural biological differences such as differences in pituitary sensitivity. The fertilized eggs appeared as a transparent whitish adhesive and 1 mm in diameter. Eggs were hatched within 25 to 30 hours. The newly hatched larvae with egg sac remain inactively in water surface. Then after absorption of the yolk sac, approximately after the 3 days, they were reached to the bottom of the harpas. They were fed with blended egg yolk and paramecium. They were reared up to the shingling stage and they were released for the brooders were collected. The place. In the conclusions, the best hormone for successful induced breeding came with the ovulin at the sex ratio male to female 2:1. This is adhesive eggs came with the size of 1 mm in diameter, hatched within 2 to 3 days. Within these tools, we are one step closer to ensure these little fish swimming in Sri Lankan rivers to generation to come. For the future studies, recommended that conduct the breeding trial with combination of the hormones and to do cryopreservations. These are my references. Finally, I would like to thank the Center for giving me the permission and support, as well as staff members of Faculty of Applied Sciences, Rajata University of Sri Lanka, for giving me the support. Thank you for giving me this opportunity.
Thank you.